yet joined, but she will join soon. So I think it is good enough for us, along with the students of our department, BSc and MSc students, and we can start with the meeting. The topic of the talk to be delivered by Dr. Subhash is COVID-19 therapeutics. So before he starts, we have some formal, say, inaugural session. And for that, we worship Goddess Saraswati. I will request Dr. Ramesh Mishra, please play Saraswati Vandana. Next to that, without say passing time, I will request Engineer Ramesh ji, please play Kulgeet. That is our culture, and it keeps us reminding about our duties and the responsibilities of university members. Please, Ramesh.
मिले हैं सत शिव सर आपको अनम्यूट करना होगा सर, अनम्यूट नहीं किया आपने।
Next to that, I request our VC sir, please bless us with his few words of blessings on this occasion.
Thank you, Sharon. My team of Department of Microbiology, led by the Captain Rajiv Gaur, and other colleagues of the department and the university. They all are good researchers and are stuck at the moment, courtesy COVID-19. And it's a really happy moment that one of your senior friend, a good researcher and academician, now residing in the very hot spot of US, will talk to you about the phenomena of COVID-19. I am a social scientist, so I can be only a victim of that rather than knowing the nitty-gritties and intricacies of that, except that yesterday evening from 6 o'clock for about an hour, the Bharatiya Shiksha Mandal platform organized a talk by the Vice Chancellor of King George's Medical University, Professor M.L. Bhatt. And there I, of course, heard in detail about, rather than the intricacies and the design and the whole phenomena, what I was able to understand properly was as to why it did not hit India that much as it hit the other parts of the world, particularly the developed world. And one of his hypotheses, based on the data that they are collecting right now, was the Indian food habits were probably the first reason which saved you from the pandemic, the extent to which it could have hit you, like other parts of the world, particularly your spices and your habit to eat something different, which has a lot of immune power, which we may not be knowing consciously, but it is part of our food. He particularly mentioned the ginger, the garlic, and the black pepper, which is a normal source of our spices in the food, as 3 prime walls against the possible attack of and the complete takeover on your body by coronavirus. And then he mentioned about the other normal habits of Indians for improving immunity. One of them, of course, is that we all played in on Earth. Never been protected from Earth. Rather, we have been part of it. To our habits of modern morning adulations have helped us. 3 good number of people get up early, so they have the first sun rays available. That is also came very handy. All these put together, and then he mentioned that your eating habits help you, and they will help you. Don't remain empty stomach. Have a good food in the morning, and what to take, what not to take. And he was a core radiologist and a radio oncologist talking down this. And he said, please depend as much on Ayurveda as you can depend on allopathic. Ayurveda is coming more handy than our medicines. We can save your life, all right, but the Ayurveda can keep you happy and healthy. And then, of course, the third Ministry of Ayush in India has also issued an open direction to everybody what to take and what to avoid during this entire period from alternative therapies. Say, for example, yoga and your pranayam, which helps your lungs. Then, of course, the kadha that they're talking about, they have especially prescribed, and a homeopathic medicine called arsenic album, which is now out of the market right now in India. It has been sold and it has been exported like anything. It has come very handy to us. So these are some of the reasons why we need to find Indian answers also as to why mortality rate is sub 3%, and if you go an absolute number, it will be 0.5% or so. And why the mortality rates were very high on some of the most developed healthcare systems in the world face the problem. Where lies the problem? You can blame, of course, that the corona that came to India was a little milder than that, but that may not be the case. Your own immune system has helped you better than the Indian habit. You can see, Subhash, it must be turning night there, but Indians by and large remain happy. You know, that happiness that we derive out of nowhere is something that keeps you very positive, and the positive energies coming in your body help you come through many of the problems of the world. I was going through, we organized one small workshop in KGMU about 3 months back. And when senior doctors came from Harvard also to participate on the theme was that how to inject Indian systems into modern medicine system. And that was the whole theme that we discussed for a whole day. And the Harvard Health Institute is doing very serious work on yoga and pranayam to understand its real impact on the stress-based diseases. And they have found some wonderful results there. So now this holistic health that we used to talk about earlier is again back in the scene as a positive answer to some of the problems that we are having right now. Microbiologists are the experts of this. They would understand it better and probably help us understand better. But only thing is that let us find an Indian answer to this global phenomena, and it must stand apart. And you should have a credible answer as to why and how Indians, without changing their value patterns and changes, systems should be able to cope up with this. My best wishes for this wonderful talk. I will miss this simply because I have 3 more engagements right now, one after the other. But definitely, the recording will be there. I think Ramesh will be recording it full. Yeah, it can be circulated and put on our website for other students and teachers to download and follow. My best wishes to Dr. Subhash Verma, Dr. Shailen, Dr. Rajiv Gaur, Neelam, and the host of other participants who are taking part in this. Let's enjoy this talk.
Thank you, sir. Thank you very much. Now I request Dr. Subhash to please take the charge and tell us about the evolutions of the COVID-19, how it has been evolving since its discovery in 2019, and how we can cope up with it. So the topic of the talk exactly is the evolution and the therapeutics of COVID-19. Welcome, Dr. Subhash.
Thank you, and I'm honored to be here to talk about the work I've been doing, and it's glad to be talking to my old alma mater, which is my hometown as well, right? So I'm excited to talk about the work I have been doing, and I can see some faces here. Neelam is right there. I can see her there, and Tuhina, I know she said she will be here somewhere. I see her there. Who else I know here? Gautam is there. So all right, let's start it, and I will let me start sharing, I guess, right first.
Yes.
So do you guys see it on your screens now?
Yes, it is all visible and good.
It's visible. Okay. Oh, not here. Where am I? Here. Oh, let me go back. It was somewhere. Okay. So thanks for the introduction. And everybody can hear me well? Okay. Right. Okay. So the topic here is COVID-19. Of course, this is very relevant because we know the entire world is going through this crisis. Mr. Trump likes to call it like invisible enemy, as you must have heard in the news and all, right? So this is a virus, which you can't see it, but it does affect our life, and not just like as a health, but also economical. Like we know that we have so much problem in the economy because of the COVID-19. So we got to find answers and come up with some therapeutics to control it. So before I start, I thought that I will show this picture because, like I said, I am one of your alumnus from RMLA and from class of 1997, which is like quite some time ago. And I was from the second batch of microbiology, which Dr. Gaur Saab, he was my master's thesis mentor. I mean, he probably still remembers my time there. And you can see him there. He
is seen here.
Yeah. So this picture, yeah, I still keep with me, and I cherish that. Yeah, I was part of this class, and I am connected with many of the students or many of my classmates, many of my friends. So I still communicate with them. And yeah, it's fun.
It's memorable.
So yeah, so a little bit about myself. I think, like I said earlier, I am a local boy. I am from Fatality. I did my master's. I'm a bachelor's and a master's from same university in 95, 97 respectively. And then from there, I went to Banaras University. I did my PhD. And from there, I went to Florence for a research fellowship for some time, like short duration. I did part of my thesis work there and then came back and defended my thesis in BHU. Followed upon, I came to University of Pennsylvania and I started working with viruses in 2001 or 2, somewhere up there. And since then, I have been working with viruses. So my training officially is in microbiology. I never changed the field. I work with bacteria in BHU, and then I started working with viruses here at UPenn. And then I came to University of Nevada, Reno, School of Medicine in 2009 as an assistant professor, and then moved up to the rank to associate professor. And then I became the director of CMB graduate program, which is Cell and Molecular Biology graduate program. It deals with admissions of PhD students and all. And this is beautiful campus of our university. You can see clearly that this is a nice, clean campus, and this is surrounded by mountains all around it. So it's a nice small town, but we have everything here in this town, like the nice university. It's fairly good ranked, and yeah, I like it here. Okay, so so when you have any questions or something, how are you going to do that? You will probably ask at the end, right? Let's keep going, I guess, right? Selen, can you hear me?
Just a minute. Yes. Yeah. Please.
Okay. So as we all know, coronavirus disease or COVID-19, it is a mixture of two words, like coronavirus, that's COVID, and disease is associated with the virus. And since this started in 2019, we call it COVID-19. And there are other names to this virus called novel coronavirus. Initially, that's how they found it. Like it was different than existing coronavirus. So they called nCoV, 2019 nCoV. And recently, which is, well, not recently, quite in March, the official name for this virus came as a SARS-CoV-2, like severe acute respiratory syndrome coronavirus 2, and SARS-CoV-2, that's how they call it. And previous outbreak of this virus was like SARS and SARS-CoV, you can say SARS-CoV-1, and also Middle Eastern respiratory syndrome virus, so MERS. The SARS and MERS were from before, and they were not that much problematic, even though they were there and they caused death, but they were not that problematic. And why they're called coronavirus, probably you know, but I want to reiterate that they have a crown-like glycoprotein, like here on your left. These are the glycoprotein, and they are like crowns, so that's how they are called coronavirus. And this is the schematic or cartoon, or you can say a visual structure of virus. That's how it looks like. Viral nucleic acid is packaged inside the virion, and there is an envelope. So it's an envelope virus. I don't know how much you know about the viruses, but There are different kind of viruses, enveloped, non-enveloped, RNA, DNA, what have you. All those different kind of viruses are out there. So the outbreak of coronavirus, I probably this started in 2019, but previous outbreaks happened in 2003. That was SARS, and that started in China as well. And that was not as much problematic here. That the number of cases were close to 8,000, not more than that, and the number of deaths were around 774 people that died out of SARS-1. And MERS was the second one, which had outbreak in 2012-13. And from that, there were limited number of cases. I guess that's 2,000 cases worldwide, and there were approximately 8,000 deaths. And with COVID-19, you can see here the number. There are around 7 million people. They are infected, are laboratory confirmed infection, and there is approximately 400,000 deaths, means 4 lakh deaths out of coronavirus. And USA is the majorly hit country on earth. We have around 2 million people who are positive or who were infected, and about 110,000 people that died out of coronavirus, unfortunately. But again, what can we do? We are trying to do every bit to save people, but it's just unfortunate that some people, they can't make it. Okay, so how do you track it? Where the numbers come from? Probably you already know this website from Johns Hopkins Public Health Department. They maintain the repository or the number tracking. And on the left, you can see this shows total number of confirmed cases. On this column, all these countries, if you click on any country, you will see the exact number of cases or approximate number of cases. And if you go on the right side, it tells you how many people died and how many tests were done. And importantly, if you look at this, you see the US is totally red. That still shows that number of cases were quite high in the USA as compared to like Europe was quite a bit. Here you can see this area, this is quite red as well. And red means the number of cases. India is not that much so far, but I think its numbers are increasing quite exponentially recently, which is unfortunate, but hopefully this will not go for too long. It will be contained. South America is having same kind of a situation. Numbers are increasing. And the worrisome part is here on this side. You can see this how exponentially the number of cases are growing still. Like this is worldwide number. So the numbers are not going down. We need to bring this curve down so the number keeps going down to the side. So, but again, it might take some time before we get there. So one thing you might be wondering, like why there are so many cases of COVID-19? There were previous outbreaks of COVID, but not too many cases were there, right? So why there is so high numbers? So one thing about this virus is that this is highly infectious virus. You must have seen that, or must have been recommended by health officials that you got to put on face masks to cover your face, eyes, nose, just because air droplet containing the viral particle can get in your respiratory system and your lung can get infected. So the infectious dose of this virus is very low. If you have an infectious particle in the range of 1,000, 500,000, people can get infected. But other viruses that don't have that high infectious dose. So that's why this virus is slightly different. And also, one term called reproduction number. That means an infected person, how many that person can get infected from that person. So for COVID-19, it's around 2.8 people can get infected from one person. Although it's not that high as measles and smallpox. Probably you know what these measles are. They're also virally caused disease, especially measles. It's highly infectious. Infectious dose is in the range of 10. Like 10 viral particles can infect a person. So it's quite high up there. And the fatality rate here, I mean, fatality rate for means the number of people die out of infection is not that much. COVID-19 is approximately like 3.4% or estimated to be 1%. So these numbers are still like settling in. We don't know the exact number because the reported cases are not exact yet. So it's quite lower than Ebola. So you probably know what Ebola virus is, right? This is another deadly virus, causes hemorrhagic fever, and people's brain, they just rupture and then the bleeding happens and they die. So the Ebola is the most deadliest virus so far. It's very, very deadly. So that's why death rate is approximately 50%. Like out of 2 people, 1 is going to die. I mean, it's so high. MERS is quite high too. Smallpox is quite high. But again, we don't have smallpox cases anymore. As probably you know, we have a good vaccine for smallpox, and smallpox is eradicated from the face of Earth. We don't have any smallpox cases on planet Earth, in any country, I believe. I mean, there are some stockpiles and what have you, but they're not there for, I mean, in actual infectious doses. By the way, can you guys see these slides? You can clearly follow the slides. Is it good enough or? Okay, all right. So you keep hearing about this term, quarantine. So what does it mean? Why this is required? Because quarantine is one of the aspects. That's how you can control the number of infections, the number of people getting infected. As we know that reproduction rate is quite high for this virus. So that's why you got to contain yourself, quarantine yourself. If somebody gets the symptoms, they should not go out in public and expose other people at risk, because those people will be infected, they will infect further, and the number will go exponentially high. So there is a term called flattening the curve. You must have heard this term as well. So what it means is, like, you need to decrease the number of cases which is exponentially going up. So you got to make sure that there is social distancing, following all these physical measures like putting masks on, that hand washing to make sure that you're not touching your nose. Even if you're touching, you're not infecting yourself. So this is important, because depending on the facility in the country, like hospital setting, number of beds, you got to maintain the number of cases or severe cases below that. Because if you don't maintain below that, the people who are not getting care, they will unfortunately die. So that's why we got to maintain the numbers lower than the capacity of the countries. That's why the flattening as a number could vary from different countries. Like US could have handled more cases. Other countries are less resourceful. They might have to have lower number of cases to maintain the healthy population, the death rate lower. So that's what this is called, like flattening of the curve. So the symptoms, I think this is pretty obvious. You must have heard or seen, or even on the advertisement or CDC website, if you go there are few things which are very particular about this virus. So the high fever is one criteria. Like people have a fever in the range of 102 or 103 Fahrenheit. So that's consistent. Besides that, coughing. People have problem in coughing. They will be coughing quite a bit. And shortness of breath, that's another thing that is critical. I mean, that one of the features or one of the differential diagnoses that physician can pick out that this person may have COVID-19. So people who have COVID, they breathe maybe like more than 30 times a minute. A healthy person that breathes like around 21 times a minute. But people who have shortness of breath, they breathe more, like they huff and puff, like keep doing those something. So those are the criteria how they are classified into COVID symptoms. And this slide talks about differential diagnosis, like if you have flu or allergies with COVID-19, if you have fever, then there is a high chance that you have COVID-19, and if there is other like itchy eyes and all those, that could be because of allergies and all those. But even in case of fever, there could be shortness of breath. So that's definitely COVID-19. But if you don't experience shortness of breath, there could be because of something else, like maybe coughing or some other common cold viruses there. So it's a sort of guidelines, but again, every person could have different kind of symptoms when they feel this or they have this infection. So again, if somebody experiences one of those symptoms, go see your doctor and make sure that they do the diagnosis or they do the test to find out whether the person is having infection or not, and based on that, they can get further like quarantine or self-isolation and what have you. So once the person gets infected, how quickly disease progresses to different stages. So there are stages where, like stage 1, during early infection, virus goes in the lungs, there's replicates, and there will be dry cough, diarrhea, maybe headache, and those kind of things will be seen. But over time, when it moves to stage 2, the shortness of breath start to set in. And person who has COVID-19, they will start to feel shortness of breath. And when it goes to stage 3, that's when the host inflammatory response kicks in, meaning our cytokine gets made. And you must have heard this term, cytokine storm. In COVID-infected people, there is a production of a lot of cytokines. The cytokines are defense mechanism to fight off certain infections, but it doesn't do very well for the body because they make a ton of cytokines. It's not specific. They go on to attack every organ, and there is multi-organ failure, and all those kind of things happen. So that happens in stage 3, and at that point they have acute respiratory disease syndrome. So they're the classic way of transition from just shortness of breath, or basically coughing, to shortness of breath, to the acute respiratory disease syndrome. So it takes around like 3 to 4 weeks before a person can experience that kind of a severe symptom. So it's a slow process. I mean, not very slow, but it takes like 2 to 3 weeks or longer sometimes in some people to experience those symptoms and can have bad consequences. So one of the symptoms which is seen in these people is, you must have heard of lung edema, right? Because that's one of the organs which gets badly affected by COVID is lungs. And as we know, we have 2 lungs, and there are alveoli. And on this side, you can see it's a normal, healthy alveoli. And what it does, I don't have to bother to tell you guys, just you see here, the blood vessels are there touching to the alveoli, and there is exchange of oxygen, carbon dioxide. And that's how this can function properly. The exchange of air and carbon dioxide comes out. But when the infection happens, this virus sets in these cells, which is lung alveolar cells, and then viruses start to cause inflammation in these cells. And that leads to secretion of these fluid, or cough, you can say in layman's terms. It goes on to fill these alveoli. And in mild cases, fine, people can clear this virus. When you cough out, it will probably come out. But when it becomes worse, to where there is an entire alveoli filled with these liquids or this cough, they cannot clear out. And that's where the severity starts, because that's when these people have to be put on ventilator because they need manual assistance in breathing. They can't breathe by themselves because their lungs are so fully filled with these protein-rich fluid or cough. So those are very difficult situations, and the stats are there. If people put on ventilator, the chances of making them out is like 50%. Not too many people can come out of ventilator as healthy. So it's like a bad situation at that point. But again, it's just one of the situations which is happening quite a bit. So, all right, we talked about the symptoms and all, and then I will tell a little virology of the virus, what this virus is, and where this belongs to. Maybe some of you are virologists, you understand virology. So there are a family of viruses, like this virus belongs to Coronaviridae, and this is grouped with SARS and MERS. And again, the corona is based on the fact that the spike protein, they have a corona-like spike, and that's how the name comes from, coronavirus. And these are around 120 nanometer particles or virus particles. And if I think I should put this in perspective, our air droplet, when we cough out, those are in micrometer range. So each droplet could have thousands of viruses, because these viruses are so tiny, they can sit on these droplets when we cough out. And when they spread in the air, they can carry in the hundreds or thousands range of viruses, and that's why when you inhale it, you might get infected with the virus. So that's why face covering or physical distancing is very critical to not get infected. And one term you might have heard about, zoonosis or zoonotic viruses. So this term means virus had jumped from animals to humans. So this is zoonotic virus. As we know that it probably came from some animal in China. We don't know exactly. Maybe it's bat, maybe it's pangolin. We don't know. There are a lot of research ongoing to figure out what had happened and how this virus evolved to become this pathogenic. So that's a question of debate, and a lot of government, like US is trying to fight, and China is trying to figure out like what has happened in Wuhan Institute of Virology, or Virology Institute of Wuhan, whatever that the name of the place is. So in terms of the nucleic acid, it's a positive sense RNA, and probably you know this term, positive sense, negative sense. And these RNA are, they can code for protein, and there are different mechanisms how they can be coded to proteins. So again, I'm not going to go in their detail, probably a little bit later on, but not here. And genome size is approximately 30 kilobases. So this is a little larger than normal RNA virus, like HIV is 10 kb or something like that. So these are like not that small. It's like a larger size RNA virus. And that is important because the mutation rate of this virus is slightly lower as compared to HIV, which is important, because if the mutation was so high, it might cause more problematic disease settings. So it's good that mutation is not that high, but it's still the virus is mutating. You guys are okay? Okay.
Fine, please.
All right.
Yeah.
Okay, so the virus, as I mentioned earlier in the other schematic, the virus, this is schematically presented how the spikes look like. So the spike is on the side here, and they attach to the cell, and that's how virus can get in. And the viral genome is put inside here in this part here. This is just to show how the virus looks like in a cartoon format. So here, this is the life cycle of the virus, because if some virus gets in the cells, what happens? Like how this virus is amplifying and making more virus. So in this case, if you see the virus is here, it attaches to the cell, which is this rectangular area, and this binds to ACE2 receptor right here. And this is the kind of lock and key mechanism. I don't know whether you know about entry of the virus, how this gets in. Essentially, what needs to happen is virus need to engage in a certain kind of a marker, a receptor, or certain kind of a key where it can enter itself in the cell, and it goes inside the cell. It can uncoat because it comes as a vesicle. Vesicle need to uncoat, and uncoating leads to formation of the genomic RNA, which is positive sense RNA, and this goes on to translate, and with translate makes these different, translate and transcribe. So with transcription, it makes these different RNA, and they can make different proteins. And these proteins, they assemble on endoplasmic reticulum. So again, this is virology, heavy virology. Like viruses can assemble in different part of the cells, like ER, Golgi. A lot of viruses, these cell organelle, right? Golgi apparatus, endoplasmic reticulum, mitochondria, right? So this virus particularly assembles meaning all these proteins, they come together and make a particle, an infectious particle, when these proteins come together, and then this buds out or goes out to produce more virus. So that's the life cycle. And this is important because if we can block any of this step, like the transcription we can block, or egress we can block, we will have a drug. So that's why I'm showing this thing. There are multiple ways we can block this virus. So there are a lot of studies going on to block the entry of the virus. Like here, the vaccine is based off that. Oh, and research is going on to block the transcription of the virus. Research is going on to block the protease function of the virus. So that will be an effort to come up with antiviral, which can lead to like not make infectious virus, and virus can die down in the cells if it goes into the cells. Okay. So here is the genome. I think in this one I just showed the overall, and here's the genome of the virus. It's around 30 kilobases long. It encodes for this polypeptide called ORF1A. Open reading frame 1A, 1B. And these proteins, these polypeptide or protein, makes multiple non-structural proteins. And this NSP12, like NSP means non-structural protein 12, is code for RNA-dependent RNA polymerase. So probably you know what these polymerases are. To make a copy of any DNA or RNA, you need a polymerase. Because for DNA, for replication, you need a DNA polymerase, because you need to copy the top strand to make it copy. It's complement a daughter strand of the DNA. So in this case, RNA-dependent RNA polymerase takes RNA as a template. Probably you know what template is, right? So the RNA is there, it binds to that, it makes a copy of that, it makes multiple copies of the virus, and that can go on to make the protein. So this is one of them. And spike is right here. This structural protein. Spike is the one which is important for attaching the virus to the host cell. So these two proteins I'm showing because we are interested in these two proteins. There are so many other proteins, they do other functions, but our focus in the lab is these two proteins. So coming back to the evolution part, so I told you about virology and what happens in the virus, but the theories of evolution, like how we believe that this virus has evolved from live animal. So you must have heard or read in the news or find news outlets and channels or whatever, online news outlets saying this virus came out of a wet market in China. Somebody who went there and ate live animal or soup of live or animal, whatever, right? And the virus was there, it wasn't totally killed, and it jumped onto human. And it was presumed that it was bat. The virus came out of somebody was making bat soup or something, whatever. And that soup somebody drank or ate, and it jumped to humans. But there are other theories, like it's not new. Jumping to different animal is old phenomena. This can happen, like this has happened previously also. Like the bat virus can go to birds, and that can go to fish and other animals. If people eat those, they might get infected. But in this case, suspected hypothesis is that this virus jumped from bat to humans because of probably eating, and from there on it went on to spread in hospital setting or in social settings, and that's how this reached to this point. And here is the slide which shows suspected source of origin. So it's like more mechanistic. So the virus is here, and what happened was that the primary host, it seems like it was bat, but there is still some question. It seems like there is an intermediate host, how this virus jumped from bat to human. So because when people sequence, or when scientists sequence this virus, they found that the virus sequence exactly doesn't match whatever is there in bat. There has to be some intermediate host, how this virus is so fit, or how this virus is well adapted for human population. So the current hypothesis is that the virus jumped to pangolin. Here, this intermediate host is pangolin. This is killed animal, and from there on, it somehow went to humans, and in social setting, it's spreading like crazy, like going everywhere. So that's the current hypothesis. And there are recent papers published in Nature and all those shows that why they believe this happened through pangolin. So what they did is they sequenced, or they matched the sequence of these different bat coronaviruses, and this is similarity match. So if you look at that, I mean, straight line will be good match to Wuhan strain, or the one which was in Wuhan, the first isolate of the coronavirus. And anything going here, any color, is showing all these different strains or genotype of coronavirus. So if you look at here, there are RATG13, this had the highest similarity to Wuhan strain. It was like 96%, 96.5% similar to Wuhan. So it seems like this virus is coming from here. But again, there is some differences here, especially in the spike area. So this is, again, the coding sequence. And here is the spike region. There is a difference that doesn't match with this even RATG13. So there must have been some intermediate mutation or some kind of a changes. I mean, there are multiple theories. I'm not going to go into that because it's highly controversial. People think that this virus came out of lab because somebody was culturing this in the lab and it's adapted. I'm not going to go get there because that's highly controversial. Government is trying to figure out why and how this virus evolved. But based on the fact, what is published, that's what I'm telling here, that there is a difference in the spike protein here, and highly similar strain is RATG13. And again, in this cartoon here, this red is the spike protein. That's how it binds to the ACE2 receptor, which is shown here in blue. And this fits so well. So, I mean, very well, matches so good. That's why virus is easily getting in the cells. And that's the reason why the virus is so infectious compared to other viruses. And here is more model, or more detailed model, how this ACE2 protein is receptor from cellular side. And this is the protein structure of a binding domain to ACE2, and the blue is the one which is highly similar to the one we have, and this is SARS-1. So SARS-1 doesn't match very well, but if you look at this is totally like parallelly matching. That means the match or ionic or hydrophobic bonding is quite strong, and that's how it can attach to that. And here is the similarities of SARS-CoV-2 and RATG13, very similar. If you look at the pattern, I'm not a structural biologist, I guess, but you can look at the color and what kind of pattern they form. They are very similar in terms of that. So there is high probability this virus had acquired somehow this RATG13 sequence, and that's how it's more virulent. Okay, so what is going on with this virulence and change in sequence? So this is the RNA virus I told you, and RNA viruses, they have tendency to mutate quite a bit. So there is a global effort by this website called, NextStrain, and somebody is interested in this, following this strain variation in different countries. They can go on. This is free of charge service. Anybody can scan the sequence, even an Indian sequence, American sequence, European sequence, anywhere. You just go and click on the side. There are multiple drop-down menus or buttons. You can analyze these sequences, how they're mutating. So these are clades like 19A, 19B, 20A, 20B, 20C, and depending on their color, you can classify what clade they belong to. So it seems like in India, this blue color are this Asian Euro-Asian countries here, right here. They have strains from all the clades. That's what it means, right? But again, you can analyze if you're interested in whatever way you want. This gives you a flexibility of looking at point mutation and what have you. All those kind of things. It gives you an option to analyze that. So the point of this slide is here to show you that every country has these different types of clades, and these are evolving. They're continuously mutating, and they're acquiring these mutations, and those mutations are being carried over, and they may be more lethal or less lethal. We don't know that answer to the question, but there is mutation happening constantly in this virus. So that was background for what we are doing. So I mean, you probably know a lot of background about COVID already, but what our contribution is in this field is we are trying to do molecular epidemiology of the virus, like how this virus is spreading. And our goal is to look at like locally. We are looking at the sequences in Nevada patients. In our state, we collect the sample, our MP swab sample, and take them to analysis to see how they track and what kind of source they got, like molecular epidemiology. And that is important because they may have some mutation which could be important later on when the vaccine comes out or when the drug comes out. So that will be predictive whether the drug is going to work, because if there is a mutation at a particular site and that's where the drug binds, the drug might not work on those patients. So this is a tracking or surveillance of COVID-19 patients or sequence mutation to figure out what is going to happen or what we are looking at down the road when this virus is persisting for long enough. And other interest for our lab is to figure out why certain people have worse outcome, and why people have like severe lung edema, or why people dying. I mean, those kind of things we are interested in. We are working on pretty interesting things, which I think is still not in publication, so that's I'm not going to talk about that. But we know that there is strain variation in some people, and that might be contributing. So again, that's not published yet, so I'm not going to emphasize more. But our interest is to figure out what is the contributing factor. I mean, not just us. I saw one paper submitted like yesterday or this past week, where they say that there is a blood group is contributing to the severity of disease. Certain blood groups are more prone to have this severe disease. And not just blood group, but there are proteins which are involved in deciding the blood group. So it's like a highly molecular complex mechanism is going on to figure out why certain people are more prone to develop disease. And that is important to know, because once we know that, we can have some kind of interventions to cure that, right? And we know that. That's why in research, we got to figure out the cause first. Then we can come with the medicine, right, or drug. So we need to figure out what is going on with those people. That's one of the area we are working on. Again, we are working locally. We are not global. We try to figure, take the patient sample, analyze them, and go from there. And we are also doing antiviral activity of the compounds. We are screening a lot of natural compound, and I have a collaboration with one of the Indian Patanjali. So we want to test their thing, like whatever. I'm not going to talk again. It's like it's still not done yet. So, but we have the compound from them, whatever they use. Like Dr. Dixit mentioned earlier, that this Ayurveda has some compound which might have some activity, antiviral activity. So we are into that as well. We are assaying to figure out what is the exact molecular mechanism, how some of these antiviral or some of these Ayurvedic drugs, or not drugs, I should say, formulations, I should say, how they are active, or what kind of things happen at molecular level to make these formulations more effective. So again, like it's ongoing work. And another area of our work is developing CRISPR-based therapeutics. So probably you know, CRISPR is an enzyme which can cleave the recognition sequence when it binds to certain sequence where it goes on. You can design those and can cleave it. So our goal is to design some CRISPR where it can go to cleave just the viral genome, not the cellular side. So we don't have off-target effects. So those are the areas of work we are doing. And in sequencing, as you probably know, it's done from nasopharyngeal swab. So what happens is just go with a swab, collect the sample from nasopharynx, and that RNA is extracted. And RNA extraction is done by a kit called viral RNA extraction kit. It comes from Qiagen. And then you do qPCR, and we decide who's positive, who's not positive. So our positivity rate is quite down now. It used to be quite high, like 2-3% were positive among all tests done. But now we have brought down to in decimal range, like 0.1, 0.2% people, they are positive. Rest everybody is like, yeah, comes out negative, which is a good thing. But again, this positive people, they go on to do next generation sequencing. So what we do is we extract the same RNA we extract from positive people. We make library and run on a sequencing platform. We get the sequence analyzed, and we match with the already existing database. And then we generate this phylogenetic tree and determine what kind of evolution is going through these viruses. And our goal is, again, not entire virus. Our goal is to focus on RDRP, or RNA-dependent RNA polymerase, and the spike protein. Again, the goal with overall goal to figure out whether these mutations may cause problems when some of these drug comes out or some of the vaccine comes out. So that's that goal. We are trying to do the sequencing, and we will be doing the sequencing for like a year or so, longer even. Whenever patient comes in, we'll collect the sample and get their sequence cataloged, meaning we'll know that what kind of a strain they had over time, and if something down the road happens, we'll exactly know whether some drug will work or not work. All right. And here the data, what we have so far is, I'm not showing all the data, I'm just showing very brief data. And this is the mutation rate in strains in the USA, what we analyzed. And here in this side, this is a dendrogram kind of thing where it shows how they are related, they are different. But this side is more important, where you can easily follow the all you can read on this side. But here you can see we have analyzed the mutation rate in different genes, and these are the y-axis shows these genes in the virus and what mutates and what week they mutated. So we followed up for 11 weeks, and we are looking at what gene is mutating faster than the others. So certain genes are mutating in certain weeks. So we don't understand those kind of things, like why this thing is happening. Maybe it's a host factor is causing those mutations, but we are tracking it. We are trying to get the information first, like what genes are mutating and what does it mean later on. But after that, we will take them, we'll follow for longer and see whether these mutations are persisting or they're reverting and all those kind of things. We want to see how this is evolving. Like one of our questions is, what evolution is happening on this virus, and what does it mean to general virus genotype or public or what have you. Okay, so one thing probably you are aware, like detection and diagnosis, we are working not a lot on that, but briefly on that. So if you know what kind of detection system is being used for the virus, probably this slide will be very useful. So just suppose this is a virus here. If virus generates the antibody test can be done, because when virus goes in the body, it can generate IgG, immune globulins, right? So they can be generated, and they will be specific to the antigens, and they can be picked out. And but okay, here, let me ask you, everybody is familiar with these terms. I'm not being too sciencey here. I'm okay, right, Salens and Dr.
Yeah, it is fine. It is fine. And the students, they have already gone through this virology and the simple steps of diagnostics and biology.
Okay.
Yeah.
Okay, good. That's good. Okay, excellent. All right. So this antibody-based testing is pretty common. It will be popular over time when we have a nice or good assay, or a specific assay, and detection of RNA or cDNA. So this is like, we have to make cDNA to do the real-time PCR, because real-time PCR cannot be done on RNA. So it has to be it has to be made into cDNA, and can be done whole sequencing, or can be done for real-time quantitation. And there are also nanopore sequencing. They can look at the bigger fragment of the RNA, and they can decide whether there is a mutation or what kind of virus is there or not. And CRISPR-based diagnosis, probably you're aware that there is technology available. They're not approved yet, but they are coming along. What they do is they design a target, or a CRISPR, where it goes to bind to the viral RNA, or viral DNA, I should say, cDNA. And when it binds to that, it can generate color. And based on the color, they can read whether the virus is COVID-19 or not. So that's a pretty sensitive assay. You don't need a lot of material for that. And also paper-based biomolecular sensor. So this is also coming along. This is not done yet, but this is in pipeline. A lot of companies are developing prototypes. What they do is they will amplify the target and go on to fuse it with certain molecule, and then if it comes in close contact with the fluoresce, and like this detection system, because we need to make some detection which is highly sensitive and quick and least expensive, so it's affordable to everybody. That's the whole goal. So the entire diagnostic field is going in that direction, where people should be able to test by themselves. Possibly they can have a paper strip, but they can put their swab and they can test, okay, I'm positive or not positive. But again, that will take time, because sensitivity is an issue, right? So, but ultimately, as a scientific community or the push from this organization and National Institute of Health or India, everybody is trying to get to this diagnostic where that would be simple, cheap, and efficient and effective diagnostics. So antibody test, right? You keep hearing about the antibody test, right? Why this is important, and what does it tell you? So there are a couple of things can be known from antibody test, right? Because antibody, if the person gets infected, like there is a viral RNA, viral antigens are made, and after the time they are exposed, our immune system sees those antigens and starts generating antibodies. So, and first response to any antigen is IgM. IgM are short-lived, but they are made. They're first immune defense to any antigen. So they will be made, and IgGs are made after that. IgGs are long-lived. So that's why we're not testing IgM, because that might tell us, okay, person is positive. We're testing IgGs there are some tests available for IgM as well. I could not say that we are not testing, but there are some tests available for IgM. But majority of the tests are being done by IgG, because that's more reliable. And it also tells you that whether the person will be protected after that or not, because once you get exposed, you must have heard that you might not get second infection, right? So it happens because when the person gets infected, their immune system makes these IgG antibodies against the virus. And when the person sees the virus again, a second infection, antibodies go on to fight it back. They can block the entry so they cannot get in. So that's the whole idea. We need to test, like, it's called herd immunity. You must have heard this term, herd immunity, right? Herd immunity works in the same way, where everybody can get infected and they have antibodies for the virus, and they cannot get infected over time. But again, the problem with herd immunity right now is that we don't know how long this protection is going to last. We know that it might last for 6 months, 1 year, but we don't have data any longer than that. So that's another problem with this virus, because this virus is replicating so fast. So we need to do proper assay and follow up over time to see whether these antibodies are protective or not. So coming back to, I think, last part of the topic, the talk is therapeutics. So therapeutics means treatment, and treatment could be either in terms of antivirals or in the vaccines. So antivirals are the compound or drugs which can block the enzymatic activity. So our goal is to have something which can block RNA-dependent RNA polymerase, just because this is an enzyme which can make the virus to multiple copies. So if you block that, you will have a good antiviral because the virus will not amplify and will die down in the cells. So that's one area we are working on. And vaccine is the other approach where you can see this cartoon here. This is spike protein, and the blue here is the ACE2, and that's how it binds here, and it can get in the cell. Fuses there, makes the lock and key, and virus can get in there. So if we have some antibodies which can bind to this spike protein, and it doesn't let the virus to bind to the target, so that's a vaccine. The virus cannot get in there because it will go and bind to saturate those sites of the virus, because virus is not going to go in a large quantity, right? It will be going in 1,000 copies or whatever copies, right? 1,000, 2,000 copies. And we have that much antibodies circulating in our system, which can go on to bind to all the spike protein. And if they bind, they will not be having access to get in the cells. And that's what the goal of these IgG antibodies or vaccines. And there are RNA-based vaccines, DNA-based vaccines. There are like over 100 clinical trials going on right now to get these vaccines, and these are different targets, different epitopes of the spike protein. And everybody's trying like different ways to come up with these vaccines where they can protect the entry or block the entry of the virus into the cells. So, one part that we are also involved here is this Convolution Theorem. So you must have heard that people donate blood. People who recover, they donate blood, right? So that once they donate blood, the serum is isolated, and serum is full of antibodies, and those antibodies are protected. So what happens is that person gets infected. When they recover, their blood is taken out, their antibody is purified, and if there is a need, they can be injected into a person who is severely sick, like for therapy, or even for prophylaxis, meaning people who are going to be exposed with the virus, like people who work in a hospital setting where they see these patients every day, like prophylaxis, meaning as a preventive measure. So people, the doctors or nurses who are going to see the patient, they should probably have these antibodies, so they will, even if they see the virus or the body sees the virus, it will not infect them. And in our area, in our hospital, what our department is doing is that we are trying to titrate the amount of antibody in different people, because that is important. We need to know how much antibodies is there in individuals who recovered. And that is also important for telling whether the person will be immune to reinfections or not. So those things that we are doing like a surveillance basis and getting those data to understand what will happen over time. So briefly before we go to other antiviral, I wanted to show this hydroxychloroquine and azithromycin. You must have heard this was a highly controversial topic some time ago, like a month ago or so, where hydroxychloroquine was approved for emergency use. Like this is a repurposed drug. Primarily, this drug is given to people who are who have malarial drug, right, or autoimmunity disease. So this drug is given, but this was effective in case of COVID as well, because what it does is the chloroquine goes in the cells which are infected, and it transfers to the lysosome. And when the virus comes inside the cell as an endosome, this fuses with the lysosome to release the viral particle or viral RNA. But this hydroxychloroquine going there, it reduces the pH, or increases the pH, sorry. It increases the pH of the lysosome, and then increasing the pH reduces the lysosomal function, so virus cannot come out of those vesicles. The virus is trapped, and that's how this hydroxychloroquine worked at the time. But again, this is not a specific drug. There are people having heart problems when they were given this drug. So it can work, but it's not for everybody. The person needs to be monitored when they are given this drug. And other drug you must have heard about this for COVID-19 is remdesivir, right? This drug is antiviral. This was approved for use in Ebola. So this drug got repurposed to be used in COVID-19, because both for RNA viruses, and they both use RNA-dependent RNA polymerase. So apparently this drug can bind to RNA-dependent RNA polymerase of COVID-19, and this can block the transcription. So here, this is the prodrug, meaning the drug which is given to people and converts to the active molecule. And this active molecule goes on to bind to this RDRP, which is here. This can bind to the active site, catalytic site of the protein, right? Protein has a catalytic site, binding site, all those domains. But this remdesivir, this is a drug which can bind to the active site and block the function of that, block the function of transcription, so there is no virus particle made. But again, this drug is not highly effective yet. The clinical trial data came out like a few weeks ago. It shows that it's not highly effective. Some people are responding very well, but not everybody is responding very well. So there is a need. There is a requirement for better antiviral, and a lot of companies are doing that, and we are involved in some of the antiviral assays as well. So what we are doing is we have developed an assay, which is a simple assay where we can screen multiple compounds by 96 well plate and even in a larger format. So what we do is we grow the cells on a 96 well plate. We add the virus or our reporter system, how we need to track it, and we add the compound in there. And what that compound does is it blocks the activity of RDRP. And if it blocks the activity of RDRP, we can assay, we have a readout way to test it. So the activity of the luciferase will go down, and that can tell us, or it can give an indication that particular compound has an activity which can block the RDRP. So based on that, we can screen, and once we screen, we go on to test on actual virus, because we cannot test this on actual virus. By growing virus is tough. It's a BSL-3 agent. I guess you know what biological safety levels are: 1, 2, and 3, 4. 1 and 2 is easily manageable, but when it goes to biological safety level 3, it's really difficult to grow in any lab. There is a special permission required to grow this virus. So that's why a screening method where people can test, we can test that what compound will be effective, it can go on to testing of actual virus. And the last one is CRISPR-based. I think many of you know what CRISPR is. It can recognize sequence. So we have designed an adeno-associated virus, which can target specific areas of the genome, and it can block, it can knock down the virus. So if the virus is not intact, it's chopped up into pieces. It will not make the transcript. It will not make the protein. So that's the goal is to deliver this intravenously or through aerosolization. I don't know that those things are later issues, but our goal is to have some kind of a AAV virus where it can go on and infect those, go on those lung cells, and the virus is there, it can cleave the virus or clear the virus. So that's the overall goal. But again, this is like a high risky project, because I don't know whether people will take adenovirus in the cell in the body or not, but at least for a philosophical point of view, this could work, and there could be variations where this may be important in controlling the disease by COVID or SARS-CoV-2. And with that, I would like to thank my every team or collaborator, Dr. Panduri. He is the director of public health. So what he does, he gives us the sample, because patients come to his clinic or his facility and get all those from him. And Dr. Rossetto, she is a section professor in the department. She is involved with sequencing. And Paul Hartley, he is director of Nevada Genomics Center, and he does all the sequencing part, and we analyze and get from there. So we also have bioinformatics part involved. I did not put them here, but they are analyzing the sequence as well. And this side is our lab. Not everybody is here anymore. I mean, some of them are gone, but that's our lab. And one thing I wanted to mention is that if you have a time to look at this video, we did this renoviral. So there is a tweet called This Week in Virology. This is a professor from Columbia. He does this for every virologist. I mean, not every, but whoever he meets. He's right here, Dr. Racanolo. He goes and he discusses about their experiences, what kind of work is going on. Well, he happened to be in our university, and we gave this interview. And I'm talking about this because I mentioned about how I got into microbiology. I did mention about our microbiology time in Faizabad and how this all happened. I did tell them how I got into microbiology and what because he asked me, how did you get into microbiology? I said, okay, yeah, this is how it happened. So, I mean, I cherish it. I like it. I mean, it's good. And yeah, it was a good experience. And yeah, it's all great. And it's a long video, though. I will have to tell you that this is a long video. This is like over an hour video or something, but you will get to know what we do. And this was before COVID. This was recorded in December of 2019, or even earlier. It was posted in December 2019. So these are my lab member, or not lab member, but department folks, and we had this interview with him. So with that, I will stop here, and I don't know whether you have enough time to ask questions, but I am here. You can go ahead and ask. Yeah, well.
Yeah, definitely. There are few questions from the audience.
Okay.
Yeah. So one question was there from one of our colleague, Dr. Manikant Tripathi. He wants to know, Dr. Manikant Tripathi, he wants to know what will happen if a recovered person is reinfected with SARS-CoV-2.
Yeah, that's a interesting question. And if the person gets reinfected, and the individual had the antibody, and he gets infected with the same virus, nothing will happen. It will not cause any severe problem. But if the person gets infected with different genotype, where the antibodies what the person has made or has in the body, it's not going to block the virus, then the person the second time, a reinfection, will have the similar problem, a similar kind of a lung edema and all those kind of things. So it all depends on what kind of antibodies are there in recovered people or recovered patients. And that will be interesting because, you know, the virus is mutating, and it's complex. It's very complex because you don't know whether all people will be protected or some will be protected, but it's a question we'll find out over time only. It's not, yeah, when we start seeing more cases and all. It's just on a hypothetical basis, they should be protected, but some are not being protected. I can tell you that, unfortunately.
Another question, curiosity from one of the student is that which cells get infected? Though you have already told this, which cells get infected of COVID-19? Yeah.
So yeah,
alveolar
cells, exactly.
Yeah.
But the thing is, the receptor, it all depends on receptor, wherever the ACE2 receptor is. And ACE2 receptor is in kidney, in heart. That's why some people are getting heart problem as well, kidney problem as well. So, but the primary route of infection is through air, right? Through aerosolization. That's why our lung cells get our upper respiratory tract, our lower respiratory tract, our lung cells, they get infected. But if it goes in blood, it might infect other organs as well, wherever the receptor is. So it all depends on lock where it snaps in and transfer the virus. Yes.
One more interesting question is there. Will COVID-19 ever be fine?
Fine?
Fine means whether will it get over or it will remain forever?
That's the question of like many people. I think this is
the
problem. And it seems like I'm not being negative, but it will be hard to get rid of this virus because so much out there. It will be really, really tough to totally eradicate this virus. So yeah, that's the unfortunate thing. But I mean, we have to deal with it. We have to just, I don't know. We have to come up with vaccines, antivirals. It's one of those questions. I think it will be tough. I will not say that it will not totally go, but it will be tough to eradicate this virus.
Yeah, one more question is there. Can we say inactivate or kill the virus by using the ubiquitination process? The spike protein, particularly. Yeah.
But I mean, you could essentially, any modification will do it, but how are you going to go about it? The problem is that how do you know it will be specific?
Yes, targeting is.
Yeah.
So one more question is there, which was asked way back. What is the mechanism to isolate protein and their effects against human? Though it is very long research question, which he has asked. So against human and how to know their genetic effects regularly change itself. Question is not very clear, I think.
No, I
What
got
is
it.
mechanism?
No, I got it. So the way you do it, go by unbiased approach, right? So if you want to know the function of any protein, you put in
Yes.
the cells and do a transcriptomic analysis and see what is changing versus wild type or control. And that can give you an idea what could be happening in the cells. That will give an indication. And based on that, you can modify wherever you want to go. Because, I mean, certain mutation might cause certain pathway to be activated, certain pathway to be inactivated. So it has to be global and unbiased approach where the protein need to be put in the human cells, or right human cells, I should say, alveolar cells. It's to go in there and analyze the pattern of a transcriptomic, an entire gene expression, a protein profiling, and see what is happening. And that would be the way to, because you can predict by computer, AI, artificial intelligence, but those are just prediction models. They're not like actual, they don't always say the actual data what happens in the cells. You can predict, but again, you've got to get into actual human cells like wet lab to get the actual data.
Right. The bioinformatic analysis requires to be proved with the wet lab experiments.
Exactly.
Yeah. So something about vaccine. This is one of our previous say dear friend of the classmate. Manoj is asking something about vaccine.
Something about vaccine.
Yeah.
So vaccine is a hot topic right now. As you can probably know, I mean, you probably know there are multiple clinical trials going on. They are in phase 1, phase 2, currently, yeah, just phase 2, no phase 3 yet. So you know how these things are done, right? When they develop, or these companies or these labs, they develop some target, which could be a vaccine target. So they put in preclinical studies, right? They get all the information in lab settings, and then goes to a small number of people, like 50 to 60 people. They inject the target and look at the antibodies, whether they're made or not. So at that point, we do have data from phase 1 clinical trial from a couple of companies. Like Moderna has it, and Inovio is doing it, Novavax is doing it, and one company from Oxford, probably you guys know, they have collaborated with Serum Institute of India, I think, right? I think that's what they, I mean, I
Yes,
find
yes,
them.
yes.
Yeah, so they are trying to do these clinical trials and they are doing phase 2, but they're already expanding those processes, because once the data comes out, they need to produce in billions of doses, right? Because so many people need it. So that's why they are preemptively going on to make more of the vaccines, whatever they have. Although their clinical trial is still going on, they don't know the exact data from phase 2. On phase 2, some of them they know, but not all of the companies they know. But I'm optimistically, I mean, I'm hopefully optimistic here, I could say. I'm optimistic that it will happen. Vaccine will come out, but might take years, 8 months, 12 months, year, I don't know. But it will come out. Vaccine will come out, but how effective that vaccine will be, that's a question still need to be answered, and only time will tell. Because but again, the good point is that there are so many companies that are going after vaccines, and they're not going for the same target. They're going for different combinations, and that will probably make an effective vaccine. So I'm very optimistic that we'll have something in terms of vaccine. But again, the time-wise, I don't know the timeline, how long it will take.
That's fine. It will require some time, and till then we need to keep safe ourselves. One question is there. Subhas, I think you won't get disturbed if we are asking so many questions.
No, I'm totally fine. I'm good.
Okay. Actually, this all is the curiosity of our students, so it is better to resolve it, right?
Yeah, sure.
One question is there. COVID-19 have direct or indirect impact on our immune system?
Yes. So yes, directly. So what happens is when virus goes in there, it can trigger our cytokine. I think I did mention that it can trigger the activation of cytokine pathways, and a lot of cytokines are produced, and that's what causes cytokine storm. So that's one way to look at it. And other way is like these proteins, they they go in and they are immunogenic, like envelope protein, glycoprotein, nucleocapsid, all these are immunogenic. So they do modify our, they are seen by our immune cells. So the people, I mean, the thing is like why older people are getting more problematic or more severe symptoms. What my belief or other scientists' belief is that people who are older, they might have weaker immune system because, you know, with age, immunity wanes down, right? It's not as good. That's why these people, they are not able to clear the virus. So definitely it has a direct impact on the immunity. Does it answer the question or no?
Yeah. One question is there, how does COVID-19 infection lead to clot in body?
Yeah, that's a seen in only a few patients. I mean, it is observed phenomena, but maybe it goes on, like I said earlier, some people who had these specific blood types, they are having more severe symptoms. So they're changing the RH factor or what have you, all those kind of things. And that's what's causing this clotting of the blood. And it's not very well known phenomena, but this is observed in some patients. Not a lot, but this is observed. Like some people are getting multi-organ failure. You must have seen Kawasaki disease and all those kind of things are happening. So this is all like area of research, but it seems like this alters the outside of the RBC or red blood cells, and that's how the clotting happens.
Yeah, one question is there. COVID-19. Just a minute. Clot का question हो गया. Yes. How do we come to know? How did we come to know that the coronavirus comes from VAT? So this is the evolutionary relationship to be studied.
How do we know? Oh, yeah. So essentially, what they did is they matched the sequence. The bat, whatever bat sequence available there, and whatever sequence of coronavirus available, and they matched. I showed you the similarity match, right? Where they look at what kind of sequences are changing, and based on that, there is no perfect match here with any bat virus. But majority of the similarity is one of those RA-TG13. That's what the majority of the similarity is. So it seems like virus must have come from there, but there is still some mutation happening. Virus adapted to bind to spike protein, because if that comes in, it will not be very, the affinity of the binding of the spike will not be as good if the mutation did not happen. So it did acquire some mutation, but the closest relative of Wuhan strain 1, or the initial strain of coronavirus, is highly similar to the bat virus. And that's the way you can say, there is no other evidence, because nobody's going to go back and look at that, or how this happened. There is no evidence, right? I mean, even if there was evidence, they will not be available anymore to look at that. We want to find out how this all happened, but how to go about it? Like, there is no traces, maybe. I mean, that's why Chinese government, they need to investigate, or they need to find out how exactly this happened, whether this is from lab or whether this is from wet market. We don't, I mean, there's theories out there, but again, I should not go in there. But those are highly political questions, right? There is a lot of money involved on that. but
So
based on similarity, I should say. Yeah.
Yes. Another question is that whether we can acquire it through the vegetables or the fruits.
Okay, so that's a good question as well, I would say, because this virus can infect our gut, our digestive tract as well, right?
Yes.
And the reason for that is our gut cells, our intestine, they express the same ACE2 receptor. So some people, they do have diarrhea-like symptoms. So it can be acquired through fruits if they're contaminated. Although it might not directly go to the lungs, but when it goes to the gut, from gut, it may go to circulation in the blood and go to the lungs. So there is a high possibility. But yes, the answer to the question is yes, it can be acquired by any contaminated food.
Yes. So I think one more question is there, and I think this should be last question. What is cytokine storm? So this has already been answered. I have one question that, yeah, if a person is already infected with poliovirus and survived, and if unfortunately it becomes infected with COVID-19 also, then what will be the survival chances?
Yeah, so there was some evidence. There was some study which showed that people who have polio vaccine or polio, I mean, virus is rare in India, I guess, too. Like we don't have any polio cases in India anymore, right? We eradicated polio from face of the earth, I guess. Some part in Pakistan, I guess. There is some cases.
Actually, the viewer has this question linked with the report which states about the similarity of the polio virus with the COVID-19 genetic.
Yeah, right. So I'm getting to that point. So there is some evidence that person might have some protection, but we don't know. There is not 100% protection. It might get lesser severe symptoms, but they will have symptoms. There is not high similarity in terms of the antibody response, which can protect the person from having severe symptoms.
So, Vas, can we see your face? Stop sharing the screen, and I think this was the last question which we had to take.
Sharing. Okay.
Yeah, because it is good to see face to face. So, any more question? If it is to be asked by someone, you may please interact with Subhash.
So is it the sharing gone, right? Sharing is
Yeah. Now it is fine. Sir. Now we can see you.
Disabled, sir. Disabled.
Okay. All right.
So it was a nice talk. Now I will request Dr. Tuhina to please complete the formality of saying vote of thanks. It was pleasure of us to have Dr. Subhash with us. Doctor, your voice is not coming.
Who's?
Verma, Tuhina Verma's.
Okay.
Yes. Dr. Tuhina, your voice is not audible.
Unmute.
No.
Still, we can't hear you. No, still can't.
Dr. Tuhina, will you please just take out this microphone wire and then reconnect it? Probably there is some technical problem.
Man. So I hope it was clear, Sarendra.
Yeah, it was very nice and very informative. Dear means though we had number of lectures also, but this was really very good, very good.
No, so I did not want to put too much sciencey thing because I thought
exactly. You know the students or even the general audience, they understand better if you put it in a more say practical way.
Exactly.
And that was there. So less science just to energize others to do science.
Yes, exactly. I mean, they need to understand the overall global thing, what's going on, right? So,
Dr. Tuhina ji, please, we are not listening you. I don't know why.
You please pay the thanks.
Sir, but she is already saying something, but we are not able to listen.
Nobody is.
Okay, so I think I take this privilege now, and I thank Dr. Subhash first. He joined us in this night time. Probably it is the bedtime for him. And
10 p.m. right now.
Yeah. And I thank you, Gaur sir. I thank Dr. Neelam Pathak. She joined from the Department of Biochemistry. Dr. Neelam Yadav, she is there. And more importantly, our group leader, Vice Chancellor sir, and the Director of Institute of Engineering and Technology, Professor Ramaparthi Tripathi ji, along with his team, Engineer Ramesh Mishra and Engineer Paritosh Singh, who provide us the technical support in organizing this Zoom meeting. So it is really, I'm very much thankful from our department that we could organize this event with the help and support of the IET team. And I thank all the audience. Please keep in touch, and more webinars to come in future. Thank you all. Thank you.
Thank you.
Okay, Ramesh ji, I think we can disconnect.
I can leave,
Okay.
right? I
Okay,
want to say
Subhash.
thank you.
Yeah.
Thank you
Okay,
right
bye.
now. Serena, thank
Oh,
you. And
okay, bye.
who else is there? I don't know anybody else. I guess. Where is it? Manoj? Manoj was there, right?
Yes, yes, Manoj is here. Manoj is here. Actually,
Oh,
he.
hey, Manoj, how are you?
Ramesh ji, wait for minutes.
Yeah, wait a little bit. I want to talk to my
Okay,
old friends here.
मनोज जी,
जी
हाँ, आपकी आवाज आ रही है, आप बात तो कर लीजिए सुभाष जी से।
जी 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 बहुत अच्छा लगा, वेरी नाइस।
Thank you. और मनोज कैसे हो भाई?
अरे एकदम आई एम फाइन, बहुत मस्त, बहुत अच्छा लगा।
Okay, good.